So, in the second step, which is second step of transcription, which is called elongation. Once the RNA polymerase has synthesized a short stretch of RNA, about 10 bases, it shifts into the elongation phase. So, this is the second phase of transcription. During elongation, as the name indicates, the enzyme performs the synthesis of RNA molecule in addition to a number of impressive tasks which are performed by these RNA polymerase. And these tasks include it unwinds the DNA in front and re-anneals it behind. So this is the complex formed. So it unwinds DNA in front of this complex. Here the DNA unwinds, and when it passes through this complex, so here it re-anneals, that is, it winds again. So this is an extra task in addition to the synthesis of RNA strand. Secondly, it dissociates the growing RNA chain from the template as it moves along. So, along this template DNA strand, here is the synthesis of RNA strand. So, this RNA, as it is synthesizing, it is attached with the DNA template. So, this in the second phase, the second task is to remove this RNA from this DNA template. Thirdly, it performs the proofreading function. So if some wrong base has been misincorporated, that will be proofread by it and it will be removed. Recall that during replication, in contrast, several different enzymes are required to catalyze a similar range of functions. That is, proofreading performed by different, unwinding is performed by different enzyme, extension is performed by different enzyme. But here, all these tasks are performed in a single step by this enzyme. The third and last phase of transcription is termination. Once the polymerase has transcribed the length of the gene or genes, if more than one gene is to be transcribed, it must stop and release the RNA product as well as dissociating from the DNA itself. So this RNA polymerase should stop synthesizing the strand, and it removes RNA. It should remove RNA from the DNA template, and it should also itself remove from the DNA. This step is called termination. In some cells, specific, well characterized sequences trigger termination. So this termination is bring about through some specific well characterized sequences in the DNA. In others, it is less clear what instructs the enzyme to cease transcribing and dissociate from the template. It is unclear. So in this figure, you can see all the three phases. It starts from here, that is initiation. This is the DNA strand, and on this DNA, this small portion is the promoter. So this RNA polymerase, which is attaches to the promoter, and it starts initial transcription. Here you can see a small transcription bubble, and then initiation, this synthesis starts. Then it goes to elongation phase. On this you can say this is the synthesis of new RNA. This is RNA strand. So here you can see this is elongation of RNA. RNA molecule is elongated, And when gene is transcribed, fully transcribed, you can see this portion. This is RNA, newly synthesized RNA, and in the termination, this RNA is released, and this sigma factor is also released from the polymerase, and this is RNA polymerase, which is also released from the DNA strand.